Alrighty, this is a short section, so I'm actually going to combine this with the next section. So, photoelectric effect. So, what the heck is it? We're seeing the photons absorb or emit in different spectral regions, so different like areas, essentially. And so this relates to the things mentioned in 311, where you are seeing the different types of molecular motion, like the vibrational or the rotational, or we're seeing the electronic transitions that we learned earlier, the electrons moving from one energy level to another. And so the one you hardcore need to know is the electron transitions, which again we've talked about before. If an electron absorbs photons, it's going to go up an energy level, right? So the energy level increases. So for example here, and then I increase to here. And then if not me, but if the electron is releasing a photon, you are going to go down an energy level. You can go from an excited state to a less excited state, or you can go from an excited state to ground, depending on how much photon is released. So remember that. Cool. So review these equations. Make sure you can plug them in, to be honest. That's what you need. I honestly don't have much to add to the College Board video. I checked a couple days ago. The video is still up, so watch that. Now, obviously, we had the drama with the other sections of the unit, as you've seen on Discord. So, but for this one, I have nothing else to literally add. It's either from the previous unit about Unit 1 stuff, or there's a couple things that are in the College Board video, so go watch that, because I really don't feel like recording it. I'm just going to record the next section. So, watch the College Board video on this section. Okay, pausing. So did you watch 3.12's video on College Board? Because I literally opened to the next slide. I'm assuming you did, because I have nothing else to add. All right, 3.13 Beer-Lambert law. Okay, so this relates to the absorption of light by solution, as I have highlighted here. And so there are three variables we need to watch out for. So on our constant equation sheet in the gas, liquids, and solution section, I have labeled here and then labeled here. So A is absorption, as it says, and then we have epsilon, which is molar absorptivity, we have our path length, and we have our concentration. What the heck is all this? Right? Like, what does that mean? And so here is this little box thing. It's called a cuvette. And so a cuvette, C-U-V-E-T-T-E, is a little box, and you put your solution in it, and then you're going to see how much absorbs in this solution. And so this little box goes into a container, which I'll talk about in a sec, and then this little red line here, that is the path length. That is where the light is going through, how long that light goes through the cuvette. We will talk about this cuvette more in a couple moments. So you need to be able to use this equation and plug and chug depending on what values are given to you. Cool. So, what the heck is a cuvette? Like, what the heck is this machine? Let's talk about it. So this is one of those experiments we're not going to do in class because I'm not about to buy one of these. They are expensive for the... Like, I don't think it's worth it, so not going to do it. So, first, an example of what we would even do this. We can find, for example, the color concentration of food dye in Gatorade. So if there was red in it, for example, I can find how, concentrated that is in Gatorade. Another thing is finding the concentration of other compounds, depending on what compounds I'm looking for. So if I have a solution that has a whole bunch of protein or a whole bunch of DNA or bacteria or whatever, I can find out that concentration amount in that solution. So we have this spectrophotometry device that you can see in the lower right. And so you need to know how to do it. I don't have this equipment, so we're not doing it in class. But essentially we have this little machine, as you can see here. And you have this little cuvette filled with whatever solution you're trying to analyze. You put that cuvette in, cool. So this is the cuvette. It is plastic. You put your solution in there and then you analyze it. So you run through this machine. You usually start with green light if you're unsure what wavelength you should be at. So green light's approximately those nanometers of wavelength. You can change the wavelength depending on what you're trying to analyze. And so then you literally just read the results. Your results will be on the machine somehow. Cool. So you choose the wavelength, you figure that out with an experiment, or you just try different ones. Again, it really depends on what you're trying to analyze, but essentially you choose a wavelength somehow, experimentally, or a range, or whatever. So what the heck is this spectrophotometry thing? Like, how does it work? So again, this is the thing that you need to know. Now, I put 3 different drawings on here, and you decide which one makes the most sense to you, to be honest. I'm going to go through the entire process of how the machine works, so that way you can answer it on a potential MCQ or FRQ. So we have the machine, and so the machine has some kind of light, and so the light, you're going to decide the wavelength of this light. It's going to go show the different colors, right? So we have the rainbow colors, and so you pick whatever color you need. And all these examples have different color choices because, again, it depends on your experiment. So you choose your wavelength on the machine, and so that color is going to go through the cuvette. So here's the cuvette, and so here you could see that this one chose yellow. And so that yellow is going through, and then as it goes through, the light is bouncing off the various particles that are in that cuvette, and so by the time the beam comes out, it's going to be less, visible or less concentrated compared to earlier. So this was the initial that I see, and then the final result after bouncing off and, going through it, because again, you are seeing how much the concentration of something is. And so you choose a specific wavelength that will bounce off the particle or whatever you're trying to analyze, and then you could see how concentrated the thing in the cuvette is. And so here's a visible way of seeing. If I have red, it gets faded as it goes through the cuvette. So here's your path length, and then it eventually comes out like that. So the cuvette has the sample, light shines through it, you see how much light goes through the cuvette, through that wavelength, you see how much is left in terms of that light, and then you see the detector is going to display whatever results you need. So it's going to show you how much of that light did not absorb in the cuvette, and then that is translated to a concentration of the sample that you are trying to study. So that's how it works. So be sure to explain that if you need to on the test. Alrighty, so here is 2 your turns. Try them out. And then pause the video while trying that out, and then I'll show you the answer in a few seconds. Ready? Here we go. So number 1 is just a plug and chug, knowing which one is which based off, the constant equation sheet from earlier here. So just making sure to read the letters, see which one it is, plug and chug, easy, right? And then number 2, it's important because, well, think about it, right? If I have something that is a smaller cuvette, that means more light is going to shine through, versus a longer cuvette, that means more light is not going to shine through all the way. So it's going to be more faded as it gets longer. So there we go. And that is it for the unit. Wow.